Hello, my name is Iulia, and today I'll be speaking to you about sex biases in maternal lactational investment in wild chimpanzees. Lactation is energetically costly for mothers, and females must balance the extra metabolic requirements of maintaining milk synthesis for their current offspring, which we call lactation effort for those offspring, against their own health and future reproduction. Variations in lactation effort and weaning can affect the fitness of females and their offspring. Lactation effort and weaning are plastic traits, so they can vary according to several factors, including according to infant sex. In the wild, we can infer lactation effort and weaning from observations of nursing behavior. However, there are several methodological challenges to this, including that infants may appear weaned from daytime observations, but they may continue to nurse at night when observations are not normally conducted, so lactation in this case would actually be longer than observations would indicate. Or individuals may continue to make nipple contacts throughout the day, but this is for comfort only without actual milk transfer. And so in this case, lactation would have actually ended earlier than observations would indicate. So to mitigate these observational limitations, we can use fecal stable isotopes to measure lactation effort and weaning. Fecal stable isotopes are non-invasive to collect, and they show daily changes in the proportion of milk in the diet. What we specifically look at are fecal stable nitrogen isotopes, or delta 15N values, which measures the reliance of an individual on animal protein and tells us whether an infant is suckling or if it's weaned. So what we're essentially doing is, in comparing the fecal stable isotope profiles between infants and mothers, we're looking at trophic level effects, so trophic level differences between an infant who's mainly consuming an animal-based diet and its mostly vegetarian primate mother. Variations in fecal stable nitrogen isotope differences between mums and infants helps us to quantify differences in lactation effort between mothers. For same-aged infants, lower isotopic differences between one female and her infant than between another mother-infant dyad suggest that the first female is making less lactation effort. Milk would be contributing less to the diet of the infant, showing the smaller delta 15N differences, and thus this infant would be farther along in the weaning process, and its mother would have spent less energy in synthesizing milk for that infant. We wanted to investigate maternal lactational investment in relation to infant sex in chimpanzees. We looked at delta 15N values to measure lactation effort between mothers at specific infant ages. We looked at the age of the last nipple contact of different infants to estimate weaning ages. And we wanted to see the effects that these had on female reproductive output. So we looked at birth dates to establish female interbirth interval lengths between one infant and a subsequent infant. Our study site is at Ngogo in Kibale National Park, Uganda. This is home to one very large community of chimpanzees. This is the eastern chimpanzee subspecies, which are well habituated at Ngogo, and they eat mainly a plant-based diet and eat meat occasionally. I conducted focal animal as well as ad libitum sampling of infants to determine nipple contacts and collected 269 fecal sample pairs from infants and their mothers and did the analyses, stable isotope analyses at the University of Calgary and at Université du Québec à Montréal. We had a total of 33 interbirth interval lengths from 32 different mothers. So for our results, we conducted a generalized estimating equations analysis to see how delta 15N differences between infants and their mothers varied according to infant sex at different infant ages. So in this graph, what we have is on the x-axis is infant age in years, and on the y-axis we have mean stable nitrogen isotope differences between infants and their mothers. And what we can see is that infant males in the dark gray boxes have a higher difference in their delta 15N values compared to their mothers than infant females in the light gray boxes. This tells us that infant males had greater proportions of milk in their age-specific diets than infant females, and suggests that mothers of sons had higher lactation effort than mothers of daughters. We also did a Kaplan-Meier product limit estimate to look at nursing survival between infant males and infant females. On the x-axis in this graph, we have the age at the cessation of nursing, and on the y-axis are the proportions of offspring still nursing at a specific infant age. What we can see is that the light gray line, which are the infant males, individuals stop nursing at later ages than the dark gray line, which are the infant females. And so this tells us that sons stopped nursing or were weaned at older ages than daughters. Lastly, we did a generalized estimating equations to look at how interbirth intervals of mothers varied according to whether they had a son or a daughter. we found that mothers of sons, which is the box on the right, had longer interbirth interval lengths than mothers of daughters, the box on the left. In summary, we found that maternal lactational investment was greater for sons than for daughters, as evidenced by the fact that mothers had greater lactation effort for sons and that sons were weaned at older ages. And these had reproductive implications for mothers, because mothers had longer interbirth intervals if they had a son than if they had a daughter. Our results are similar to other studies on deer and other primate species, including chimpanzees at other sites. How do we explain these findings? Well, first, it may be that male offspring are just generally bigger than female offspring, and so in this case, sons may require greater maternal resources just to survive. However, studies that have looked at body size differences between infant males and infant females in chimpanzees haven't found much of a difference, and so this explanation is not as likely. We could also have something called the biologically fragile male hypothesis working here. This suggests that in several mammals, including humans, for example, infant males tend to have worse outcomes in times of hardship. So they have higher morbidity and mortality if there's an epidemic or a food shortage, for example. And so in this case, chimpanzee females may be overcompensating in their investment of their male offspring just to ensure that they survive in case there's a time of hardship. Alternatively, there could be a potential for higher reproductive success for mothers if they invest more heavily in their male offspring than in their female offspring, especially at Ngogo, which is a site with very high food abundance and high food availability, as well as low feeding competition. And so in this context, it may be more beneficial for mothers to invest more heavily in their male offspring. So this could be a strategy for mothers to maximize their own inclusive fitness, because they're able to influence the reproductive success of their sons more than their daughters, especially given the fact that chimpanzees are male philopatric, so females remain, or sons remain with their mothers throughout their reproductive lives. So there's more potential there for mothers to influence the reproductive success of their sons. Alternatively, if we look at it from the female infant perspective, there may be some benefits to attaining independence faster for female chimpanzees. So female chimpanzees have to emigrate at sexual maturity. And so if there's some kind of correlation between earlier weaning and social competence, this could be adaptive for female infants later on in their lives. So with that, I'd like to thank the following institutions and people for their support, and thank you very much for listening.